Our overall goal in the neurobehavioral core is to further the understanding of brain function, both in health and disease. We do that through basic behavioral assays in rodent models, as well as more advanced techniques that allow us to monitor and manipulate neural activity.
We can answer these questions of what is it that makes us behave the way we do. We can see what's going on with our brain activity that will produce this behavioral outcome.
We can help from project conception all the way to completion. We're continually adding new equipment and new expertise. All of the sort of major techniques in behavioral neuroscience, where we can do basic behavioral assessments, but we can also then use more advanced new technologies from electrophysiology to optical imaging approaches that allow us to actually see neurons firing.
Because a lot of behaviors have connection between each other, if we want to answer one question, sometimes we would need multiple different behavior paradigms. That's where the behavior core becomes very important and very helpful, because they are focusing on developing these different behavior paradigms.
They helped me do everything from designing the experiment to then analyzing the results. Sometimes they help even perform the experiment, so we can get a more specific and broad view of how the brain is affected when we interact with those channels.
We're able to assess a variety of behavioral endpoints in mice using behavioral assays. So these are things where we use different mazes and types of specialized boxes that we can put the mice or rats into and assess different behavioral outcomes. We're also increasingly doing this in fully automated ways. So while the mice live in their own home cages, we can do quite a bit.
We can see sleeping, eating, locomotive activity as the mice are being tracked.
This is really proving to be quite powerful. I think one of the most exciting things about it is that we're able to generate a large amount of data more efficiently and more reliably than we ever could. We're actually monitoring them continuously and also bringing in new approaches like machine vision that allow us to, with high precision, decode kind of the behaviors that the mouse are engaged in 24/7 without the need for sort of painstaking human observation and annotation.
The experimenter isn't really involved with handling the mice, so this is less stress going into the experiment in the mice.
We can monitor physiological measures like cardiovascular outputs, brain measures, all in a continuous manner. And it's sort of an approach we're calling holistic or integrative physiology, where we're able to get a huge amount of information out of mice so that we can accomplish multiple research goals at the same time and use less animals. We also do a lot of more advanced techniques where we're actually peering into the brain while they're behaving. Vibrophotometry, miniature endoscopes, wireless telemetry, so that we can get an insight into the kind of underlying physiology and connecting neuronal activity to behavioral outcomes.
We have wireless telemetry devices which can give us EEG and EMG recordings. We can see slow wave sleep 1 and 2. We can get paradoxical sleep. And we're working towards combining that with readouts of neuronal activity.
Virtual reality is a novel and interesting technique to use in mouse models. One of the major advantages is that it allows us to have a head-fixed mouse engaged in complex behavioral tasks, which allow us to use advanced imaging approaches in the brain during the behavior. We can also use it to do things that you can't do in the real world, so that we can kind of break down the rules of how the animals are sort of constructing their representations of time and space.
We can see which neurons in a particular area are firing when the mice is doing this particular behavior. And then if they do a different behavior, maybe we'll get a different set of neurons that are firing. With that readout, we can see how heterogeneous the brain is.
I think it provides a pretty unique opportunity for researchers who are interested in doing this type of research. They can just come to us and we provide the expertise and the space and the training. They can come with the ideas and we can bring, our input and kind of work together to come up with the project.
I started to work with Dr. Cushman to set up a fear conditioning paradigm. Setting up a behavior paradigm is not an easy job. It takes a lot of time. If we set them up by ourselves, we could run into a lot of difficulties to know which is the right direction.
We work pretty seamlessly with the other cores here at NIEHS, so once we've completed a behavioral study, we can take tissue and send that to a variety of different places here so that they can utilize their expertise.
The people in the group are very approachable. I feel like I can come to them at any point with questions. They're very happy to answer me, and they even ask me good questions. They're very kind and eager to partake in the science and learn.
The ability to work in an environment like this with so many amazing scientists and to just be able to focus on the science is very inspiring to all of us.
The brain is one of the most important organs in the body, and being able to understand what is going on with that will help us be able to come up with better therapies in the future, or just get a better understanding about who we are as people.